and which helps us to perceive things clearly. Similarly, when we hold a glass in our hand, it is possible only because the coordination of the sensory neuron and motor neuron which signals the muscles to hold the glass. So, number one function, the neurons are involved in reflex action. Number two, intricate connections between neurons in the brain and spinal cord. Then, so these are the two important functions of neuron. Then comes the neural conduction. Now, what do you mean by this conduction? A neuron that is at rest, that is not firing a nerve impulse or a message, is actually electrically charged. So there is a jelly-like solution inside and outside the cell, which consists of charged particles known as ions. Now there are electrical charges across the neural membranes, which constitute the positively charged ions known as cations and negatively charged ions known as anions. So both positive and negative charged ions are present inside and outside the cell. Now there are mostly negatively charged ions inside the cell and positively charged ions outside the cell. Why? Because because of diffusion, that is the process of ions moving from areas of high concentration to the areas of low concentration, and the electrostatic pressure, that is the balance of electrical charges when the ions are at rest. The positively charged ions are like, for example, sodium, that is Na plus, calcium, that is Ca2 plus, that is the positive, potassium, that is K plus, and chloride, that is Cl minus. Now there are relatively more positive charge next to the plasma membrane on the outside and relatively more negative charge on the inside of the membrane. Now this difference in electrical charge across the membrane is known as the membrane potential. So this difference in electrical charge across the membrane is known as the membrane potential. So in an inactive state, the neuron is said to be resting. That is, it displays membrane potential or resting potential. So this is known as the resting membrane potential. The magnitude of the potential difference is measured in volts, capital V, or millivolts, that is small m, capital V. And the resting membrane potential is abbreviated as capital RMP, is maintained with the help of principles of diffusion, electrostatic pressure, ion channels, and sodium-potassium pump. So in brief, the nerve impulse and it goes through the cell body to the axon to these terminal buttons. Now when there is a stimulus at the spike initiation region that reaches the threshold of exciting, it depolarizes the membrane. This leads the sodium ion to move into the cell with the help of forces of diffusion or via electrostatic pressure. This makes the inside of the membrane more positive with respect to outside for a temporary period of time, that is from minus 70 millivolt to plus 50 millivolt. So the action potential, that is the electrical charge reversal at a particular point along the axon, it reaches its peak in about 1 millisecond. So no more sodium can enter the cell and potassium ions. They start leaving the cell, and this causes the membrane potential to restore itself to its resting state. Now the action potential is an all or none phenomenon. Now such that whenever it is initiated, it goes down the axon to its terminal buttons, or it does not occur at all. So the information is sent down axons via small electrical impulses called the action potential. Action, A-C-T-I-O-N, action potentials. And the amplitude or the size of the nerve impulse, it depends upon the particular neuron as well as the rate at which the neuron conducts the impulses. Now, when the potassium ions begin leaving the cell, it causes a state of depolarization of the membrane. Once enough positively charged potassium ions are replaced or released out of the cell, the membrane reaches its resting state. Sometimes too many potassium ions, they leave the cell and thus making the membrane slightly hyperpolarized. But then the potassium channels close, making the membrane potential, it reaches its normal resting state again. So in my next class, I will start with the synaptic transmission, where the structure of synapse, the steps of the synaptic transmission, and what are the importance of this synapse and neurotransmitters. So today, first, in my first slide, I told you that in this unit, that is unit 2 of this biopsychology, I will deal with this structure and functions of neuron, classification, process of neural conduction, and the process of synaptic transmission, functions of neurotransmission, and lastly, the neuroplasticity and related concepts of neural degenerations. Okay. So, first, I told you about the Williams-Harrop and the Santiago Ramon Y. Casal's experiment on neuroscience, because he was awarded the Nobel Prize in Medicine in 1906 for the work on the structure of the nervous system. Then, what are the structures of neuron, starting from soma or cell body, axon, terminal buttons, plasma membrane, cell body. It consists of the Golgi body, the Nissl bodies, the mitochondria, rough endoplasmic reticulum, etc., and the dendrites. Now, what is an axon or axon hillock? It is a long, slender part of the neuron that extends from the portion of the cell body. It is also known as the axon hillock. There are some other primary cells present. They are the neuroglia, the glial cells or the glia. There are 4 types of glial cells, that is the oligodendrocytes, and the Schwann cells, the microglia and astrocytes. Then I told you about the nodes of Renvier and the astrocytes. Then according to the structure, the types of neuron, there are three types of neuron, that is the unipolar, bipolar and multipolar. So unipolar of neuron, it is also known as the pseudo-unipolar neuron. Then the bipolar, that means two polar, so it is known as bipolar neuron, and multipolar, that is these neurons have several dendrites, one axon and several dendrites. Now then, there are two types of Golgi, that is the most commonly found in the brain and the spinal cord. Now Golgi type 1 and Golgi type 2 neurons. Now what is Golgi type 1? They have very long axons and few branches, while Golgi type 2 neurons have shorter axons and branch they repeatedly, and these neurons act mostly locally to nearby areas. Now on the basis of the functions, neuron may be classified into afferent neurons, efferent neurons and interneurons. Now what are the functions of these afferent neurons? They carry the impulses to the central nervous system. They are affected by the changes in the environment, and these efferent neurons, they are the they takes the impulses away from the brain or the spinal cord to the muscles or glands. And lastly, the interneurons, they lie within the CNS. They carry information from the afferent neurons towards the efferent neurons. Then what are the functions of neuron? Mostly there are two important functions of neuron. They are, neurons are involved in the reflex action, all the reflex action going on in our body. And number two, the intricate connections between neurons in the brain and spinal cord. Then in brief, I told you about the neural conduction. So what is the membrane potential? What is the resting membrane potential or the RMP? What are the ion channels, etc. Now in the next class, I will start with the synaptic transmission. So if you have any question, you can please ask me. You can write it in the comment box. I will try to give you the answer. Thank you. Thank you everybody. আচ্ছা, ঠিক আছে, যদি কোয়েশ্চেন থাকে বলবেন। আচ্ছা, একটা মানে ওরা জিজ্ঞেস করছিল কয়েকজন স্টুডেন্ট আমার, যে ওরা এটা কি অ্যাটেন্ডেন্সটা দেওয়া হবে, তো সেটা একটু ক্লিয়ার করতে ওরা চাইছে। আচ্ছা, এটা একটু মেলে যদি দিয়ে দেন, ওরা একটুখানি কনফিউজড এই ব্যাপারে। হ্যাঁ, ওকে, থ্যাংক ইউ, থ্যাংক ইউ।